So let's know about the physiology of fertilization first. Conception results from the fertilization of ova by the spermatozoa. The mechanism whereby spermatozoa pass along the uterus is not properly explained. As the ciliary movement of the cervical and endometrial epithelia is downwards, the spermatozoa must migrate against the cilia current. It can only be assumed that spermatozoa, which live in an attractive alkaline medium of seminal fluid, find the acid environment of the vaginal secretion lethal in a matter of 2 to 4 hours. The cervix has the same pH as seminal fluid and is undoubtedly demonstrably attractive to the spermatozoa. Spermatozoa are powerful, fast swimmers, and from the time of ejaculation to the time of arrival in the ampulla of the tube, it takes about 60 minutes for the spermatozoa to cover the intervening 20 centimeters. The distance compared to the size of spermatozoa represents a rapid and purposeful travel. The subendothelial layer of the endometrium exhibits increased upward peristalsis during the follicular phase near the ovulation time, and this may hasten the migration of sperms into fallopian tube. It is now generally accepted that though spermatozoon after ejaculation may remain motile for a long period, its useful lifespan is limited to 24 hours. After the short interval, it is less capable of performing its biological duty. The period of survival of mature ovum is even shorter than that of spermatozoa, the time which elapses after its escape from a ripe Graafian follicle and its entry to the fallopian tube, which is potentially fertilizable, is estimated at 12 hours and rarely up to 24 hours. The significance of the statement is that coitus, to be capable of fertilization, must take place in the 24-hour period around ovulation. Ovulation most commonly occurs around 14 days before the onset of next period, though variations are known. Fimbria of the fallopian tube by muscular contraction spread out over the ovary at the time of ovulation, a movement which simplifies the transport of the discharged ovum into the lumen of fallopian tube. Furthermore, the musculature of the fallopian tube undergoes rhythmical contractions, especially at the time of ovulation. It is most likely that peristaltic contraction of the fallopian tube determines the transport of the ovum towards the cavity of the uterus. The sperm that reaches the ovum first penetrates the zona pellucida, normally inhibits entry by other sperms. By the time fertilized egg enters the uterine cavity, the endometrium has grown under the effect of progesterone into the secretory endometrium and is ready to receive the egg for implantation and provide its nutrition. On general principles, the blame of infertility should be bared between the two partners. It is not uncommon for patients to complain of difficulty during coitus when they have little knowledge of the correct method to be employed. During sexual intercourse, the erectile tissue around the vaginal orifice become engorged and the vaginal orifice become more patulous. There is discharge of mucus from the duct of Bartholin gland, which acts as lubricant. Female orgasm is induced by stimulation of clitoris, partly during penetration of penis and partly as a result of clitoris being rhythmically pressed against the penis after penetration. Importance of external genital areas of sexual stimulation must not be forgotten. These erogenic areas vary with the individual and their susceptibility to stimulation is equally variable, but their aggregate response is cumulative and plays a vital role in ultimate achievement of orgasm. The seminal fluid is mainly deposited in the posterior fornix of vagina, but it is possible that some of it is ejaculated directly into the cervical canal. It is also believed that concentration of the contractions of the uterus fallopian tubes during female orgasm cause seminal fluid to be aspirated into the cavity of uterus, and this aspirating effect is responsible, in part at least, for the migration of spermatozoa upwards into the fallopian tube. The female orgasm is not essential for conception, and it is not uncommon to see women who have conceived without full consummation of marriage and in whom hymen is intact. In such cases, the spermatozoa, having deposited around the hymen, migrate on their own motility along the whole length of vagina and uterus. Infertility. Infertility implies apparent failure of a couple to conceive, while sterility indicates absolute inability to conceive for one or more reasons. If a couple fails to achieve pregnancy after 1 year of unprotected and regular intercourse, it is an observation to investigate the couple. This is based on observation that 80% of normal couples achieve conception within a year. It's observed that 50% conceive within 3 months following regular unprotected intercourse, 75% in 6 months, 80 to 85 in 1 year. It is termed primary if the conception has never occurred, and secondary if a woman fails to conceive after having achieved a previous conception. Incidence is about 5 to 15%. Optimal age for conception is 20 to 35 years in a woman. After the age of 40, the fertility is reduced and there is increased risk of chromosomal abnormalities and other malformations in the fetus. For a man, age is less important, but after 50 years, decreased libido, sexual dysfunction, reduce the fertility and predispose to malformed fetus. Therefore, it may be prudent to proceed with investigation of apparent infertility in a woman near or after the age of 35 years. Instead of waiting for a year, if she seeks gynecological help. Conception is the result of successful fertilization of the female egg by the sperm. Hence, the couple should be counseled individually. Male infertility, spermatogenesis. It occurs in seminiferous tubules of the testis. The primordial germ cells appear in the yolk sac in the 3rd week of embryo, migrate along the dorsal mesentery to the genital ridge. The germ cells divide into primordial cells or spermatogonia by the 6th week. They remain quiescent in the seminiferous tubules throughout the childhood. Near puberty, spermatogonia divide into primary spermatocytes. Meiosis occurs only at puberty. Till now, mitosis occurs. At puberty, meiosis occurs and smaller secondary spermatocytes containing haploid number of chromosomes are formed. These develop into spermatid. The spermatozoa develop by acquiring an acrosomal cap, elongation, condensation of the sperm nucleus, and a tail. The development of sperms takes 72 days, and entire spermatogenesis, including transit time in the duct, takes 3 months. The mature sperm has a head with acrosome covering midpiece and a tail which allows normal motility. It's here the acrosome, and this is the head, the postnuclear cap, the neck, and the middle piece. Acrosome membrane contains enzyme hyaluronidase, acrosin, and other proteases which allow acrosin reaction, breakdown of acrosome membrane, penetration of sperm into the zona pellucida. Hyaluronidase dissolves the corona radiata cells. The sperms are stored in the epididymis. One spermatocyte produces 4 spermatid and one spermatid produces 4 spermatozoa. Spermatogenesis beginning at puberty is a continuous process unlike ovulation, which occurs once a month and continues with senescence though with less efficacy. The testes show germ cells in different stages of maturation at any given time. The sperms mature in the testes as well as accessory organs, undergo capacitation in the cervix before they are capable of fertilization. The seminiferous tubules are lined by germ cells and Sertoli cells lying adjacent to the germ cells. The Sertoli cells produce androgen binding protein by follicular stimulating hormone and bind testosterone to this protein, causing a high level of testosterone within the testis compared to that in the blood. Interstitial cells produce testosterone by luteinizing hormone. Endocrine control. Hypothalamus is critical in the development of male organs and spermatogenesis. Gonadotropin releasing hormone GnRH in males is produced continuously, unlike in a pulsatile fashion in females. FSH is not essential for spermatogenesis. It acts on Sertoli cells, produces androgen binding protein as mentioned above. Sertoli cells also produce Mullerian inhibiting hormone and inhibin, which inhibit FSH. MIH inhibit the development of Mullerian system. LH stimulates testosterone secretion by Leydig cells. Hypothalamic failure leads to loss of spermatogenesis and testosterone production. Spermatogonia are diploid germinal cells which divide by mitosis into spermatocytes. These undergo reduction division meiosis 1 into haploid secondary spermatocytes which by meiosis 2 develop into spermatid. These spermatids develop into compact, virtually cytoplasm-free sperms with condensed DNA in the head, capped by apical acrosome and a tail. These sperms are incapable of fertilization after they undergo capacitation in the female cervical canal. The entire process of spermatogenesis takes 74 days, and if we include transport in the ductal system, it takes 3 months. They are present in the testes in different stages of development at any given time. The testes produce 200 to 300 million sperms daily. Capacitation can also be induced following incubation in the culture media in IVF. Cervix plays following role in reproduction. It gives nutrition to the sperms. It gives alkaline medium for the survival of the sperms. It sieves out abnormal sperms, causes capacitation of the sperms, storage until upward pulsation of the sperms. Acrosome reaction is an important component of capacitation for zona penetration into the oocyte. Acrosome is modified lysosome over the sperm head. Under its action, the overlying membrane becomes unstable, breaks down and releases hyaluronidase enzyme, which allows the penetration of corona radiata and zona pellucida. The Sertoli cells line the seminiferous tubule and extend from base of the membrane to the lumen. They support the spermatid, possess the receptors for FSH and testosterone. The tropic effect of FSH and testosterone on spermatogenesis is mediated via the Sertoli cells. There are 4 sperms per a Sertoli cell. The Sertoli cells produce Mullerian inhibiting factor which prevents the development of Mullerian cyst. Sertoli cells also produce testosterone binding protein which maintains high level of testosterone within the testis. Endocrine control of spermatogenesis. It depends upon the hypothalamo-anterior pituitary testicular functions. GnRH stimulates the anterior pituitary gland to secrete FSH and LH. FSH acts on Sertoli cells. LH triggers testosterone secretion by Leydig cells. The concentration of the testosterone is higher in the testis than in the plasma. The testosterone exerts a negative feedback on the pituitary gland as well as hypothalamus. Fertilization. Following capacitation, a mature sperm meets the ovum in the ambulatory portion of the fallopian tube. By acrosomal reaction, hyaluronidase release, it penetrates the zona pellucida, which in turn prevents entry of other sperms called polyspermia. It is possible to aspirate the polar body or blastocyst cell for genetic study of the embryo without disturbing further development of the embryo.